So good afternoon, my name is Yassi, and I'm from Professor Gerson Silva's lab from the Federal University of Rio. And today I'll be presenting a little bit of a broad aspect of the physiological P53 aggregation studies in our lab, which is one of the main line of research. The P53 has been directly linked to cancer because of its activity as a tumor suppressor. And this protein activity is triggered by stress signals such as DNA damage, hypoxia, oncogene expression, and then P53 works in the response of those stress signals into such responses into cell cycle arrest, migration, and apoptosis, for example. It is a transcription factor, and it regulates genes that are involved in those responses. It's interesting to notice that P53 gene is mutated in over 50% of the human cancers. And those mutations are most frequently in the DNA binding domain of this protein. And the loss of function of P53 has been attributed to cancer because of P53 function in averting tumor formation. And that has been shown by studies that used P53 null mice, and they actually found that these mice die of cancer at a young age. So P53 mutations, they have a dominant negative effect. And the protein, the wild-type protein, is active as a tetramer. So it forms tetramers, and those tetramers are able to bind DNA and act as a transcription factor. The mutants of P53, though, they can be able to form the tetramers, but those tetramers are not able to bind the DNA, and therefore they can't do P53 regular functions in the cell. So the loss of function of P53 has been classically explained by the incorporation of the mutant P53 into the tetramers of the wild type, forming heterotetramers, and those mixed tetramers are defective in gene regulation. But more recently, our group and others have suggested that the P53 inactivation is due to its self-aggregation related to a Prionlike effect exerted by the mutant protein on the wild-type protein. So what's a Prionlike effect? Prions were first described by Stanley Prusner back in 1982, and he got the award, the Nobel Prize award for it. And prions is he called it proteinaceous infectious particle because what he noticed is that the prion protein, which is was then related to as the agent of the development of prion diseases such as Creutzfeldt-Jakob disease and mad cow disease. He found that this protein suffered a change in its conformation, and the converted protein was able to convert further wild-type structured protein into a malformed structured protein, and then that would capture proteins into aggregates and amyloid, leading to amyloid formation and ultimately at the disease. And that concept, it's important to notice that it's also, it was also described as a transmissible effect, since if healthy organisms were in touch with the converted protein, they would get the disease. They would get the conversion of the wild-type protein and then the development of the disease. And that was found to be related to the loss of the wild-type protein and then the loss of its functions in the cells. And that would cause the diseases. So this concept of prions have been evolved throughout time. And now it's actually applied to different kinds of proteins, such as A-beta protein, tau, and alpha-synuclein, that were also shown to form these aggregates, to be transmissible, and to cause neurodegenerative diseases. So what we believe is that P53 actually can fit in that group. But how did we get to that? So P53 aggregates first came up, I don't know, back in 1992, when groups were looking for patients, cancer patient tissues, and they were looking for P53, how it was found in those cancer patient tissues. And so they were really intrigued with what they found, because they not only found that those tissues had P53 mutants in it, but they also found that wild-type P53 was accumulated in the cytoplasm of cancer cells of the patients. So here's just showing one of their images. This is a work done by Moll and collaborators back then. This is immunohistochemistry. The first here panel is just hematoxylin and eosine, and showing the nuclei and cytoplasm of the cells. And in B and C, they did immunolabeling for P53, and that's showing that they actually found that the protein was being accumulated in the cytoplasm of cancer cells. They didn't really know back then if it was aggregates or what it was, but they discussed that what was happening is the tumor was, the cancer was developed because of the loss of function of P53 once it's supposed to be in the nuclei of cells, acting as a transcription factor, and so as a tumor suppressor. And so they were like, if it's capturing the cytoplasm, it's not in its site of action, so it's not acting as it should be, and therefore the cancer is developed. So from then on, a lot of works came up, a lot of studies showed, and they were finding, all of these works were finding in tissue, in cancer patients, they found P53 accumulated in the cytoplasm in several and diverse kinds of cancers and patients. So this accumulated P53 in the cytoplasm of cancer cells was further on characterized as aggregates. And our group have shown in 2003 the characterization of some of these aggregates. So by atomic force and transmission electron microscopy, we were able to characterize the aggregates under denaturing conditions, and those aggregates were found as forming granular and annular aggregates, and also under regular conditions, the most common form of it was fibrils. Later on, our group also found that those aggregates had an amyloid nature, which relates P53 a lot to prion protein that has the same kind, the converted form also is able to form amyloids. And it's important to mention that structural destabilizing mutations in the DNA binding domain increase the aggregation of P53, showing that the converted form of this protein, which would be the mutant, is actually more prone for aggregation. And more recently, our group have found some evidence that actually links P53 further as a prion, and that's shown in one of the pieces of data from this work is here, showing that the mutant P53 can be found, and it's strongly found in amyloids of tumor biopsy samples. So here, this is immunofluorescence, and it's a good tool to use for that because we have specific antibodies for oligomers that are gonna give us an idea for aggregation, right? So when you immunolabel P53 here in green, so here it's showing the wild type protein and the mutant protein. This is one of the hotspot mutations in arginine 248. So when you immunolabel P53 into these tissues, you actually can compare if it's colocalizing into aggregates. And it was found that some of the wild-type protein can be found in those aggregates, but most importantly, the mutant is mainly colocalizing with those aggregates. So as you can see, green is the mutant again, and red is the oligomer, and again, you see a huge colocalization between the mutant and the oligomer, showing that this converted form of P53 actually has this big aggregation behavior. And another way of showing you that was done by Kinetics of aggregation, and these assays are done by the use of thioflavin molecule. This molecule is able to bind aggregates, oligomers of fibrils, and and once it binds it, it's able to emits fluorescence, and that fluorescence can be captured and detected throughout time. So that's what was done here with the recombinant proteins, the wild type P53 and the mutant P53. And those kinetics showed us that the mutant P53 has a much higher propensity to aggregation compared to the wild type. It's faster and higher. So that actually shows us that the converted form of P53, which we're trying to compare to a prion conversion, is has much more, it has a behavior that tends to form aggregates, and the wild type doesn't as much. So what we were missing, what we wanted to see is if this mutant, if this converted form of the protein can convert further the wild type into those aggregates, because that's also another effect of the prion proteins. So what we did is a classical seeding experiment, which this kind of assay was first, of course, done for prions, for the prion protein, and then it's been incorporated to different kinds of proteins that have this kind of behavior. So what's done is the converted form of the protein, in this case the mutant P53, is induced into aggregation, and then a seed of it, or a tiny bit of it, is introduced into the wild type, or it's mixed together with the wild type, and then you can test if the wild type is aggregating further. So the mutant P53 aggregation is not hard to be induced. If you put it at 37 degrees for 30 minutes, you can get a huge amount of aggregated protein. And that was diluted 10 times and put together with the wild type protein. And then we did the kinetics again with thioflavin to check for its aggregation. And as you can see, the seeds were actually able to increase the aggregation of the wild type protein and made its aggregation occur much faster than its regular pattern. So far we actually have identified two important effects, Prionlike effects for P53, which is it has a converted form that's prone to aggregation, and also that this converted form can actually further incorporate the wild type form into aggregates. But what about the transmission? Can this be transmitted to other living systems? So far, we need further evidence, but it's very interesting to note that recently a group has published that cells can actually incorporate aggregates from the extracellular media. So for getting collaborators, I'm just showing you a piece of data from their work, but they showed that, so again using immunofluorescence, they were able to put mutant aggregated P53, tagged to dye-like 650. And they were able to detect that in their experiment, they put that in the extracellular environment, wasting time, and then image the cells to detect if the cells are incorporating that. It's been shown, they also showed that cells are able to secrete P53 and also aggregated P53. So here, they show two kinds of cell lines, and in blue is the nuclei of the cells. In red is the actin showing the membrane of the cells, and the aggregates of P53 with dye-like 650. And as you can see, I don't know if it's good light here, but what basically is shown here is that those aggregates were found inside of the cellular membrane. So inside of the cells, showing that those cells were able to internalize those aggregates of P53. So that's a very good insight, and maybe with further confirmation, we can actually say that P53 has this converter form being able to be transmitted between cells. And summarizing what I've said is that P53 in wild-type cells, normal cells, it's there in the nucleus of the cells, acting as a transcription factor, as a tumor suppressor. And when P53 is mutated, it loses its function, and the wild-type protein can be converted into these aggregates that are formed by the mutant P53. And that will convert further on wild-type protein, occurring then a loss of function of the wild-type protein into the aggregates. Those aggregates can then be secreted by cells and be captured and internalized by healthy cells. Those healthy cells will then be converted and suffer malignancy, and that, of course, with the loss of P53 and the loss of function of P53, will further lead in cancer progression. So that's a very interesting idea. It's a different perspective of seeing the development of cancer. And what was really surprising to us is that recently a group has showed that the mutant P53 can actually form aggregates with P73 and 63. So these two proteins, they are members of the same family as P53, which is called the P53 family. And they are very similar to P53 in structure and sequence. And what Zhu and collaborators have shown is that the mutant P53 is found in aggresomes, which are organelles in the found in the cytoplasm of cells that have a lot of compartmentalized aggregated protein. And they found that these two proteins can be colocalizing in those organelles. So as you can see here, by immunofluorescence, they did a staining for the nuclei of cells. This is just wild-type protein, so wild-type P53 and 73 or 63, the first panel. Blue is the nuclei, red is staining for P53. And as you can see, P53 is nicely spread in the nuclei of cells in its site of activity, working well, and so is 73 and 63. They're also transcription factors, and their site of activity is also the nuclei of cells. And they, of course, colocalize a little bit. They have similar activities there. But when the mutant is put into the system, what they could see is that this mutant, if you can see here in the cytoplasm of the cells, there are these agglomerates of protein, which are the aggregates of the mutant P53. And when you look for 73 or 63 in green, they're actually colocalized into those aggregates. And that was seen for two kinds of mutants that have hotspot mutations for P53, which are the arginine 282 and the arginine 110. So that's a nice introduction actually for Elio's talk. He's going to show more of our recent data from the lab and a lot of studies that we've done with P63 and 73 compared to 53. Thank you.
Thanks for your talk. So I had two questions. First one is there any specific mechanism or anything known about how or why that specific mutant aggregates? And the second question is, how does this aggregation affect cancer mechanisms, including like metastasis and other things that are different from DNA control, transcription control?
So it's not this specific mutant. Most of the hotspot mutants for P53, especially the ones in the DNA binding domain, have this characteristic of aggregation. And the mechanism, what is known is that it's because of its, well, so far our group has shown that it loses the binding of water molecules, and then it can form aggregates with each other, right? And but basically, what is seen is that it aggregates. And so you asked about the cancer functions. Yeah. So the thing is, P53 is related to several cancer responses. It's not only DNA repair or apoptosis, but it's actually being linked to, I don't know, throughout the development of cancer, P53 can act like delaying tumor growth, cell proliferation, DNA repair. So there are several senescence, there are several functions. So P53 is kind of a key protein in this process. So that's why we believe the loss of its function contributes so strongly to the cancer development.
So is there any critical concentration for this aggregation, such as the minimum proteins P53 to aggregate?
Sorry, I didn't understand.
So, I mean, is there any critical concentration for this aggregation to happen?
Concentration?
Yeah, for this.
Critical concentration. No, in vitro what we've seen is you can actually detect aggregation at any concentration. But especially for the mutant form of the protein.
Jim? Ask a question, yeah. I was commenting. Well, just regarding the amyloid part of your talk, so like peripheral amyloid, as far as I understand, is different than brain amyloid, which is a beta sheet, whereas the peripheral isn't. So how does that play into the story there? Is the P53 aggregate, is that a beta sheet formation, and is there a relation to beta amyloid in the brain? How does that work, please?
Yeah, it basically forms beta sheet amyloids, aggregates. That's the nature of the aggregates, the amyloids.
So like brain amyloid, unlike peripheral amyloid?
Yeah, more like brain amyloids, yes.
Thanks a lot, Lacey.